trials. I won't talk about any of my. I'll talk a little bit about my basic science research, but I thought this group consists largely of clinicians, so let's talk about it. My disclosures are here. I've done some consulting for private companies. I was going to talk about the Marinius trial, but we just won't have time for it. Uh, so what I'm going to focus on is what did we learn as the people who designed and executed ESETT? How did we react to ESETT? And how did we go on to design the next trial, which is called ketamine for a established status epilepticus treatment trial that we hope to start in winter of 2025. And I just want to walk you through the process of designing this trial as we did it. Just to give you a sense of how long it takes to design and do a trial, this dates back to, ESETT dates back to 2010, when I stepped down as the president of American Epilepsy Society. A group of people who were thinking of designing established status epilepticus trial, who are all listed at the bottom, asked me if I could lead the group. And if you can see I started working with the group in 2010, and we went through many steps that you'll see happen. You have to get permission from NIH. You have to get major networks to agree. Then you have to design your trial, and then you have to get FDA to agree. So it took 5 years from 2010 to 2015 to actually plan design. And this is the first patient for ESETT was recruited at Stanford on November 3rd, 2015. I never forget that day. So ESETT was a multi-center randomized comparative efficacy study of fosphenytoin valproate or LEV in ED for patients who had failed benzos. So we all know what status epilepticus is, and patients found having seizures were treated with benzos. We hoped they would get adequate doses, but many times they didn't. But after they got repeated doses of benzos, if they continued to have seizures, they were treated with drugs, fosphenytoin, levetiracetam, and valproic acid. And what we at that time designed the trial, a successful outcome was that the patient should stop having seizures and their responsiveness should improve at 60 minutes from the start of drug infusion, and they shouldn't get any other seizure medications. The idea was that if you're comatose, we are not sure whether seizures are continuing or not. At that time, we didn't think we could get EEG to confirm absence of seizures. And one of the things that we were aware, and we said that if you got intubated, that was failure, because when you do intubation, you use propofol typically in rapid sequence intubation, and that is an anticonvulsant. So anybody who got intubated failed the drug. And this is what we found. Big picture in 384 patients, which took us 5 years of planning and 3 years of execution, was that LEV was effective in 47%, fosphenytoin in 45%, and valproate in 46%. One of the things that you'll notice is that the denominator is different, that LEV got 145 patients, fosphenytoin 118 patients, and valproate got 121 patients. This is because of Bayesian adaptive design, and I'll get towards it talking about what does Bayesian adaptive design do. But in adaptive design, what happens is that patients get preferentially allocated to the drug that's performing better. So even though I was blinded throughout the trial, but looking back, at some point in the trial, the patients doing on LEV must have been doing better, so that they were preferentially being allocated to LEV. But in the end, you have reversion to mean, and adding subjects, basically we reverted to that mean. And if you look at outcomes in 3 age groups, what you'll see is that in children and adults, that the outcomes are very similar. In children, you get slightly higher than 50% efficacy for LEV. And for adults being 18 to 65, it's somewhere below 50% and slightly lower efficacy in older adults. As I said, that we were worried about intubation, and I want to talk about, before I talk about intubation, I also want to talk about hypotension and cardiac arrhythmia. One of the big drivers of this trial, especially from Europeans, was the concern that phenytoin causes hypotension and cardiac arrhythmia. And that's a black box warning on FDA label. And at least neurologists strongly felt that phenytoin causes, and it's a dangerous drug to use for treatment of status epilepticus. But in children, as you see, there was not a single instance of cardiac arrhythmia in 229, of which about a third were treated with fosphenytoin. Hypotension occurred, but no more than valproic acid. But one thing that was different, and we all debated this for a long time, and I don't know how to interpret it, is that kids treated with fosphenytoin ended up being intubated 3 times as often as those treated with valproic acid and LEV. There is no other there were 2 other open label clinical trials, one in Australia, one in England, that didn't find any difference in intubation. We have searched literature all the way back to 1930s, whether phenytoin increases risk for intubation, but this was very, very significant. There was just no doubt. As you can see, the p-value is 0.0001. So take it for what it is. We found in this trial, and this persisted throughout the trial, that intubation with fosphenytoin was much higher than valproic in children. If you go to adults, again, hypotension and cardiac arrhythmia that we used to worry about didn't occur in this trial. So if you can use fosphenytoin without worrying about those complications, though in extravasation and overdose is always a concern. In fact, we had 2 overdoses of phenytoin in children in our trial, in fact, and that was a significant cause of concern. I don't know if there's someone from CHOP, one of the kids ended up in CHOP after that. Respiratory depression, as you can see, is similar, but endotracheal intubation in this age group rises to 20%. And again, there's no difference in 3, but intubation now is about 25%. And then as you grow older, again, no hypotension or cardiac arrhythmia. I was very scared that older adults than 65 would have a pretty high incidence of cardiac arrhythmia or hypotension, especially with phenytoin, but we didn't see it. That didn't happen. But the biggest thing is, as you grow older, the endotracheal intubation keeps getting more and more. Depakote had the least endotracheal intubation, and Depakote appeared to be somewhat more effective in older adults than younger adults. The sample size, if you recall from my slide, for older adults older than 65 was a lot smaller. But if we were ever to carefully look at older adults in status, we should definitely look at valproic acid as a potential second-line treatment. So as I pointed out, when we sat down to look at the results, we were disappointed because we thought at least one of the drugs would be better than 2 others. And the fact that there was null hypothesis, some scientists would say none of your drugs work, right? Because you didn't have a placebo arm. What if we can obviously ethically not leave patients untreated, but you could argue that none of our drugs worked or all of our drugs worked equally. And so we thought, what could be driving this this result? One was that we didn't have EEG. EEG may have demonstrated much higher efficacy than what we found from clinical exam. I've already brought up intubation. And we found that we were concerned that intubation rates may have changed our outcome. And what we did find was that there are some EDs that tend to intubate a whole lot more than other EDs. And it tends to, the practice tends to vary with how busy you are. If you're in an emergency room that has overcrowded with a lot of people waiting, physicians tend to intubate because they don't want to have a patient stop breathing while they're busy with another patient. On the other hand, less busy emergency rooms tends not to intubate that often. So there is a pretty significant variation in practice, as you can imagine, that if you're intubating a patient because you want to send them to CT scanner, that's not really a failure of the drug, but it got counted as failure. And the third thing, which is my background as a status researcher, is I said, look, we were treating seizures and status epilepticus have a different pathophysiology. And our intervention should seek to break the cycle where seizures get more seizures rather than focusing on just terminating seizures. And so my argument was that anti-seizure medications were used. So we considered and chose ketamine to add ketamine to our study and go forward with ketamine. Ketamine over the last 25 years has been used a lot in refractory and super refractory status epilepticus. There are systematic reviews, case reports after case reports. More than 400 patients have been reported in the literature. I think now it's become so common, nobody's publishing that data, is just there. And people report efficacy in 20 to 60% of patients. It has a pretty strong safety record. In what we wanted to do, of course, was to bring this to the emergency department, moving from ICU to the emergency department. And we know that ED physicians use ketamine all the time to reduce fractures, for conscious sedation, for pain. They are pretty familiar with using ketamine. And one thing that they love about it is that they're not likely to intubate a patient who goes on ketamine. There's a lot of data of 10,000 patients treated with ketamine in emergency departments where they didn't need intubation at all. And that meant for us we could use a drug that was not going to cause respiratory depression and go from there. So I'm going to summarize like 20 years of my research in like 2 slides. Basically, what we argue is that when you have seizures of status epilepticus, first thing that happens is that there's reduction of GABAergic synaptic inhibition, and there are a lot of all the initial treatment of status epilepticus and refractory status epilepticus is based on just enhancing GABAergic inhibition. We give benzodiazepines after benzodiazepines, and when benzos fail, we give second-line agents, and then we give midazolam or propofol, which also work on GABAergic inhibition. Those are the sort of standard of care things. But what happens during status epilepticus is NMDA receptors get activated. They enhance AMPA receptor transmission. Activation of NMDA receptors causes neuronal loss and long-term plasticity. I'll just show you some key data on it and how that happens. How that happens is that when the seizures are occurring, NMDA receptors get activated. There is intracellular calcium entry, which activates CAM kinase 2. That causes phosphorylation and internalization of GABA A receptors and reduction of GABAergic inhibition. It also activated some phosphatases, which cause trafficking of AMPA receptors. Specifically, GLUA1 subunit of AMPA receptors is trafficked to the surface. Glutamatergic transmission shown here is enhanced GABAergic inhibition. This is synaptic currents. These are actual recorded synaptic currents, as you can see in an animal with status epilepticus. The inhibition goes down, excitation, synaptic currents go up. This is due to receptor trafficking. And our goal, I believe, in treating status epilepticus is to block this vicious cycle of receptor trafficking and we can achieve with an MDA receptor blocker. And the concept is illustrated in this slide, which I generated exactly 35 years ago. If you take an animal and just give it seizure after seizure, what happens is, this is in 6 hours, giving seizures every 5 minutes for several hours. The seizures get longer and longer, but if you add MK801, the seizures will not get longer. They occur, but they don't get longer. And same with treatment with ketamine. Here I gave ketamine multiple times, and seizures didn't get longer. And in status epilepticus recorded with the EEG, both my lab and then Claude Westerlein's lab at UCLA, both of us have done a series of studies showing that if you combine diazepam with ketamine, various doses, and we've done both diazepam dose response curves and ketamine dose response curves, you get true synergism. That is, you get an effect that is larger than either drug alone. And again, a lot of work done in Claude's lab and initially in my lab, showing that there is true synergy of ketamine and benzodiazepines. Finally, Denny Fujikawa in late 90s and early 2000s had done these elegant studies showing that if you block an MDA receptors during status epilepticus in animals, experimental animals, the neuronal loss that occurs with status epilepticus in experimental animals is protected. Also, work done by at Bertram and Bob DiLorenzo and Richmond. They've shown that if you, in experimental animals, clinicians don't deal with this. If you give status epilepticus to an experimental animal, they will develop epilepsy. But if you treat the status with either ketamine or other NMD antagonists during status, what happens is that far fewer of them will develop chronic epilepsy. So from basic science perspective, there was a very strong basis for bringing ketamine early treatment of status epilepticus. Then the question became, and the group agreed that we should try to test ketamine. But the question then becomes, what dose to use? And we worked with Lisa Poles, who's at pharmacology and pharmacy at University of Minnesota. She's an expert at pharmacokinetics. So this is a basic pharmacokinetics. Remember that we are going to give a single bolus dose. And even though pediatric clearance of ketamine is faster, it doesn't matter, because what we are really hitting is peak dose effect in the PK. Second thing was to convert all the animal work to human data, because we had done all this work. Claude's lab, my lab, had done how to convert, take that data in rats and mice to bring it to human beings. And fortunately, FDA has a guidance for industry on estimating how to convert human dose from rat dose. And we had a sense of that 30 milligram and 50 milligram kilogram, some of the doses that we had used in rats would be equivalent to IM dose of 5 milligram and 8 milligram per kilogram. And we wanted to convert that to human dose in IV, which would be 2 to 3 milligram per kilogram given IV. And then Lisa looked at what kind of effect these doses would have. So we know that for light anesthesia, you can use 2 to 5 milligram per kilogram. For dissociated state, you can use 1 to 1.5 milligram per kilogram. Analgesic doses, and these days, ketamine is in fashion for depression. You can get nasal inhalers. Those are much lower doses that people use. Patients with depression are doing. So we thought we were coming towards this dose. Then Lisa did PK modeling studies with some of her colleagues at University of Maryland, and we knew that this is the anesthetic dose. If we gave 1 milligram per kilogram over 2 minutes, we could go way higher than anesthetic dose. However, if we were going to give it over 5 minutes, which is what we were planning to do, there'll be a very brief period where ketamine 1 milligram kilogram would go higher than anesthetic. And if we gave 3 milligram per kilogram over 5 minutes, shown in this line, there'll be the peak will be clearly higher and sustained for at least 5 to 7 minutes beyond the anesthetic dose, but neither one would require intubation or anesthesia. So it would be anesthetic, but won't require any respiratory depression. So our goal is we used LEV because it seems to be the safest in children and adults. And it seems to be the most commonly used drug after ESETT, using the same ESETT dose of 60 milligram per kilogram. In ESETT, we used LEV for 10 minutes, but we didn't feel any restriction to spread it over 10 minutes because we are no longer using phenytoin. So we propose to load it over 5 minutes. This will be our control arm of 60 milligram per kilogram for 75 kilos, and others 4.5 grams for people above 75 kilos, and then 60 milligram per kilogram IV. And then we'll either give 1 milligram per kilogram ketamine or 3 milligram per kilogram ketamine. The trial is done under exception from informed consent. That is, no patient, we think nobody who's in status epilepticus can give informed consent for a trial. So what happens is the patient is enrolled and then later get consent. But any of you wanting to do EFIT trial, I can tell you that FDA has changed its rules, and FDA now says that we must say that we will try to get consent and relatives have opportunity to decline, which we always do. But finding somebody's relative while you're trying to treat status epilepticus isn't easy, but we have to say that we'll give opportunity to decline in the trial. That is, failure to decline is not consent, as you can imagine, but we have to give opportunity to decline the trial. We obviously have to do large-scale community consultation before we launch the trial, and then each site will do community consultation where they'll know that trial is about to happen, and people can opt out, and do a public disclosure also besides consultation. Public disclosure means putting ads in newspapers and letting people know through all kinds of social media, etc., that we are going to do this trial. If you have questions, ask us. And all this gets submitted to FDA as a docket. So the trial would run essentially similar to ESETT in that patients will be picked up from the field, and they're having seizures, and then they get benzodiazepines. We want to make sure that they get adequate doses of benzos. So we always say that if in doubt, give another round of benzos before you enroll the patient. The enrollment occurs, and this time we want cerebral device connected as soon as the patient gets enrolled into the trial. So we're giving extra money and extra incentive for every patient who gets enrolled, that the moment you enroll them, please put the headband on, and the only reason we chose Cerebral for anybody who's curious is because it's the only FDA approved device for up to 2 years of age. There's no other rapid EEG that is currently FDA approved. That is the only reason. And we'll ask them to, and the drug will be 5-minute infusion, and then we'll wait. Physicians have 10 minutes to decide let the drug work, because Keppra-LEV takes time to get in the brain. And so we don't want people treating and declaring it failure until 15 minutes, and then observe them for next 45 minutes to see if their seizures don't recur, they don't get intubated, they are not given any other. And the primary outcome is at 1 hour, even though we'll collect information. Randomization, again, those of you, what we do is we pre-randomize. There is a box sitting in the emergency room where we tell the person use next. What happens is there is a central system at Medical University of South Carolina which tracks every box that is used. And in order to maintain a balanced randomization, they will tell coordinator to change their use next box if they think the randomization is going out of whack. But randomization, the only thing we are looking at is in terms of stratifying randomization is by weight group under 30 kilos, which is about 8 years of age, median age of a child weighing 30 kilos is about 8. So, and the reason why we did this is we don't want any child getting overdosed. 2 of our patients in ESETT got overdosed with phenytoin. Both of them ended up in ICU for a long time. One of them was in CHOP, one was in, the overdoses didn't occur at CHOP, but the ICU was there. And now every child can only get maximum of 35 milligrams of ketamine. That is about 3 times the maximum safe dose. So we just want to prevent any overdoses that happen by mistake in emergency room. So, and there'll be a dosing chart, which is weight-based, and these kinds of charts would be available for any patient. Basically, you have to estimate the weight and give the drug. The drug volume is based on the weight estimate for children. Once somebody is above 75 kilos, there's a standard, basically the adult vial, you can run it. We are still debating whether there'll be 90 ml or 100 ml in the adult vial, because people tend to run the whole vial. I don't know why, but they take a vial of drug and they just give the whole vial, even if they're not supposed to. So, but we'll decide that. And so the maximum dose, as I just said, will be, these will be the 3 arms of the trial. And as I just said, we'll use cerebral device to get EEG data on every patient. We are all waiting with bated breath as to how high the compliance will be, because we are depending on emergency room nurses, staff to start it. It takes about 20-30 minutes for the study team to arrive by the patient's bedside. So it'll depend on ED physicians remembering to put this thing on. So, and we'll spend a lot of time trying to train them to make sure that they put it on. And of course, after it is put on, let's see how the signal quality is. We'll have central interpretation. So EEG will be interpreted by Eric Rosenthal and his group at Mass General. However, local hospitals will have access to cerebral data. So if any of the local physicians and neurologists want to look at the cerebral and make treatment decisions based on that, they'll have access to that. So as I mentioned, that we were very concerned that our ESETT primary outcome was too restrictive and it may have influenced the trial results. So there was a long 6-month debate as to what the primary outcome will be for our ESETT trial. And what we came up with was a 5-step outcome rather than a binary outcome. And what we said was the best outcome is that you stop seizing, recover, wake up, which was the same other is that you stop seizing, but you don't wake up because you have some stroke or bad thing going on, but EEG shows no seizures. That's number 2. Third is that you had no seizures going on, but somebody decided to intubate you because they wanted to intubate you for other reasons. And fourth is that despite treatment, electrographic seizures and life-threatening hypotension, those seizures continue. That is, the drug failed. This true drug failure occurred. And finally, if you have a life-threatening emergency. So how many of you think that was a good outcome to choose? Nobody likes it. So this was 6 months of work. We actually submitted it to neurology. Neurology actually accepted it begrudgingly. I don't know, Barbara Yopes must have said, okay, Jaideep's paper, I'll publish it, because the reviewers also complained about it. But this is what we came up with, but I'll show you what happens in real life when you get very creative. Then the idea was best outcome, you wake up without seizures, you're still sleeping after seizures, you get intubated, but seizures stop, but seizures continue, or bad things happen. FDA said, get out of here. You can't do this. FDA said your primary outcome is composite scale that relates to factors related to efficacy and safety. You mix safety with efficacy. This composite primary endpoint has potential to confound. That is, imagine a drug that prevents intubation but does nothing to stop seizures. Your outcome will go from 3 to 2 without doing anything to seizures. So you mixed up efficacy. They always say that if you ever are going to go to FDA with a clinical trial design, don't mix efficacy with safety. And they just said, no way are we letting you do this, at least from FDA perspective. And so we were pretty stubborn, and we went to NIH with that outcome and said, we'll see, and FDA will convince it. And we went to a NIH study panel. And of course, you have to give all of your FDA documents to NIH study section as well. Just be aware if you ever do this. And they slammed us. They gave us a score of 45. They said, this is not, the first thing they said was, nobody's using ketamine in pre-hospital. And then they said, vague definition of primary outcome, need better justification for response rates. 5-tier outcome is not clinical. So FDA doesn't like it. So they gave us a score of 45. So with both FDA and NIH telling us to go back to the score, redesign, we said, okay, we give up. We went back to the ESETT outcome. And we said, okay, ESETT, you all accepted, NIH accepted, New England Journal accepted. So we'll go back and use that, but we'll use EEG because both FDA and study section had said that use of EEG to determine outcome is better. So basically we said you have to improve consciousness with no seizures or no or absence of seizures on EEG monitoring. One thing we had to guard against was that we know that some fraction of sites will never have EEG because they forgot to put it on, or they recorded bunch of garbage, bunch of artifact, and EEG reader says this is uninterpretable. So how do you guard? This was in order to guard against that, what we said was, if the EEG is absent and patient doesn't wake up, we'll consider it a failure. How much contamination occurs because of that? What it is we know that 47% of patients in ESETT woke up and had no seizures. So that's 47% don't need an EEG. 25%, as I just showed you, get intubated. So EEG doesn't make a difference once you intubated the patient. So that 25 plus 47 gets you up to about 70-some%. So the only remaining 28% of patients who are not waking up and not intubated, where EEG will be critical. And even if we miss 10 or 20% of our EEGs, miss that, our flaw rate will be 2.8 or 5%. So that's how we thought through EEG, because we remain worried that people will fail to record EEG, or EEG will have problems, but we remain to see. So this is just a flow chart. Primary outcome, success if they're awake and not having seizures, that's fine. Again, if they're intubated or not, if they're intubated, EEG is not necessary, but if they're not intubated and not waking up, EEG will help us decide whether they have electrographic seizures. The EEG reader aficionados, all those of you, we spent a lot of time discussing what is electrographic status, because if you look at ACNS criteria for definition of status, they said 20% of EEG must be dominated by seizures for 60-minute EEG, but we are getting EEG for only 45 minutes after treatment. So we're taking 20% of that, that is 9 minutes of seizures in the 45 minutes following recording, or whatever, 20% of the recording. Now, some people would say any seizure following treatment should have been considered treatment failure, but after much debate and argument, we picked 20% of the record as the definition. And I'm sure people will comment at the end of this trial, whether it was a good choice or a bad choice, but that's how we made it. Primary outcome, I've told. Secondary outcome, we still want to do our door outcome. We are in love with it, so we'll do it as a secondary outcome. Then we'll look at a bunch of safety things like who gets intubated, how long are they in the ICU, how many people die, the usual stuff. Now, because the design is dichotomous, I just want to walk you through another thing that takes a long time in clinical design, because we do it as a Bayesian adaptive design. So for those of you who don't do clinical trial design, the classical way in which people do clinical trials is that you take one treatment, you take standard of care, like many of us do anti-seizure medication trials, take a refractory patient either gets randomized to treatment or drug, and then we say, 6 weeks or 6 months, is there a 50% reduction in seizure frequency? What is the proportion of patients who have 50% reduction in seizure frequency? That's how we do it. It's the sample size is decided ahead of time, and you assign essentially the same number of patients to both arms. And this is classic, what we call frequentist design for Bayesian, for clinical trials. The Bayesian design, what it allows you to do is that you look and in middle of the trial, not the team, I'll be blinded, but the statisticians look and then decide how the trial proceeds. So it's like Bayesian design is like if you live in Southern California, and most days it doesn't rain, so you don't take an umbrella with you. But one day you get out of your house and you see overcast sky, the probability that it'll rain and you'll need an umbrella is high. So you'll take an umbrella with you. So you add one bit of information. So you take extra information, what they call if you're a statistician, you get your posteriors there. So if a drug is performing better, then you allocate more patients to that drug. And as you heard in ESETT, we allocated more to LEV because LEV seemed to be doing better. And that adds power to the study design. In fact, ESETT, which was meant to be a 5-year, $20 million trial, we finished it in less than 3 years for less than $15 million because we had an adaptive design. In 3 years, the biostatistician said we are 99% sure that there is no difference between these drugs. There's no point in enrolling any further subjects. And that's the power of a Bayesian adaptive design. So we're doing this again with Bayesian adaptive design, but with how you go through the process is first, our mathematical hypothesis is that at least one of those two doses of ketamine, 1 or 3 milligram per kilogram, will improve outcome in 15% of the patients. So bad outcome is 53% from ESETT. Subtract 15% from there, so it's 0.38 is what we will hope to get with at least one of those two arms. And then question is, what is the maximum sample size we'll need? And when do we do adaptive randomization? So basically, Jonathan Beal at Medical University of South Carolina, along with Valerie Tarkovsky, they did series of simulations, and they do simulations like these to say, what if we were to do what if we were to do burn-in period of 350 subjects or 400 subjects, 350 subjects, or 425 subjects. That is, the first time we look at the data is this, and then do a sample size of 650, 700, 750, 800, 850. If both the drugs work or only one drug works, what is the probability of our type 1 error? Just remember, this is type 1 error, one-sided, but we want both-sided alpha. So if you look at this table, the type 1 error goes below 0.25 at a sample size of 700 with the first randomization, first response adaptive randomization. That is, first look at that data at 350. So in this scenario, we enroll all subjects up to 350 subjects, and then start adaptively assigning more patients to the better performing arm after 350, and we'll have an alpha less than 0.05. But second consideration is power, whether we'll have more than 90% power to detect differences, or what we call beta. And this, again, we could see that with a sample size of 700, we could get power in above 90% in most scenarios. So obviously, we want to keep the sample size minimum. So what we'll do in this design is we'll do 1-to-1 randomization for first 350 subjects. Then we'll do early stopping, just like we did for ESETT, if we know for sure that there is a clear winner, or if there is a futility. If neither stopping criteria or are met, then we keep assigning patients to 2 active arms in proportion to their likelihood of success, and we repeat every 100 subjects, and we stop at 700 regardless. Though the probability that we'll need all 700 patients by our simulations is pretty low, but that's the outer extreme for this trial. And these are the stopping criteria for this will be debated with FDA. This may be debated by FDA. We are hoping that they are strict enough that FDA won't object to it in our final submission. So we'll have 22 hubs and from Siren and 17 sites from PCARN. Total sites we expect to be about 60. In ESETT, we enrolled about 16.66 patients per month, which should take us 54 months. That is 4 years and 6 months to get to the better end of 700 patients. Because of adaptive randomization, we hope that we can end the trial early. So in September of 2023, FDA thought our revised primary outcome was okay. We submitted our grant in November. We got a review back in just this April. This time NIH gave us a good score of 25. Counselors recommended funding. NINDS has run out of money, so they say they'll give it to us in December. And so hopefully we get the money in December and start working. And these are all the people who did all the work in designing it, but I'll stop sharing and answer questions.
Thank you very much. That was a wonderful talk, and interesting data that you presented, and also the new study. It's very exciting. I see that Pete has his hand up. Pete?
So that was great, Jaideep. Are you going to AES this year? No, I'm just kidding. So are you, I wanted to know, so reading ESETT, I was always curious about this. So as you study status epilepticus going forward, does etiology matter? So when you think about status epilepticus and developing broad therapeutic strategies, does Norse versus Dravet versus hypoxic ischemia versus cardiac arrest, does it matter, or do you think it doesn't matter?
Beyond this trial, it definitely matters. I have a painful job of writing a review article off and on. I honestly believe that perhaps the last time we can treat all status as the same is when we give the first round of propofol and or midazolam, perhaps. And I wish there was a good study to prove or disprove it, that regardless of whatever your etiology, you can get away with treating all statuses the same. But I think first time you fail those anesthetics, we should be thinking very, very seriously of etiology. And I think, and that's the challenge of designing ICU trials. I was going to talk about, when I said beyond, I was going to talk about ICU trials. And I honestly deeply believe that once you fail one round of anesthetic, deeply thinking about etiology is very important. Otherwise, we are just doing the same thing.
Yep. Thank you. Great talk, Jaideep. Thanks very much.
Jenny?
Marana, you had your hand up first.
Oh, I'm sorry.
It's fine. Thank you so much for the wonderful talk. I had a question. My question is about interaction between lamotrigine and ketamine. How much do you think it's like a concern for you at all? Is this something that you're going to look into, given the effects and the complications? And are you going to exclude them, measure the lamotrigine level and the onset?
What's the interaction? Let me confess, you're the first person to tell me about it. Nobody's brought this up, so educate me.
So this is, so I know about this because Dr. Dusseldorf at Stanford, we do a lot of ketamine studies on our intracranial patients, and he always sends us these emails, stop all lamotrigine, don't resume the lamotrigine, because he does ketamine studies on them. So there is interaction between lamotrigine
So what happens?
and ketamine.
Lamictal levels rise?
No, what it does is lamictal is going to decrease the effect of ketamine, and it can
Is
also
it PK or PD effect? Do you know? I'll have to read about it.
Well, it seems that it interferes with glutaminergic neurotransmission. That's why, like, kind of reducing the ketamine effect. I'm not sure, but he knows what he does. Like, he's the guy behind the optogenetic.
I need to learn more.
Okay.
We didn't find in ESETT, we looked at the home drugs and effect of home drugs. Question was whether if you're taking Keppra, does Keppra work as well for treatment of status or not? And it was almost statistically significant, but not quite. And it didn't really matter which drug you were on in terms of giving the same drug for treatment of status.
Okay.
But lamictal, I don't know. Thank you, and I'll investigate. None of my pharmacology colleagues pointed this out, so thank you.
Of course.
Yeah, so my question, Jaideep, was about just how other ketamine studies have informed your trial. I mean, I believe that there was one that was ongoing at Alabama at one point. I don't know as much on the pediatric side, but how did those issues inform your trial?
So yeah, that helped me a lot with the study section because they wanted, so there are 3 studies, one in Italy in kids and adults, where they enrolled subjects much like ESETT, and there are 2, one in a pre-hospital where they're giving ketamine right with benzos. And of course, these are open label studies, and everybody got 23 out of 24 patients were cured. You know how open label studies reports are. So yeah, they use between 1 and 3 milligram per kilogram. So when I was writing the grant, there were 24 patients reported in early treatment. I'm hearing anecdotally from my ED colleagues that some EDs are going rogue and using ketamine even in the rescue squad without any evidence to support it. And there was long debate whether we should even use ketamine even before in pre-hospital setting. But given the worry about intubation, especially with 3 milligram per kilogram, I could convince Robert, my colleague, that we should do it in the ED. So, but I'm hearing rumors that some rescue squads are doing it. Who's next?
Is it Taslima? Oh, I think you muted.
Yeah. So my question is that I wanted to ask for ESETT, I was just curious, why was lacosamide or Vimpat not included? And in general, do you feel that Vimpat can be used successfully for status epilepticus? I know we often use it once we're running out of options or if one of the other ones are
Yeah.
contraindicated or not available.
Yeah, so we and if I can tell you, others will criticize us for not using phenobarbital, and we had to pick three because every time you add an arm, you're adding 106 somewhere like 200 subjects to your study, and it gets more and more expensive and more less and less feasible. NIH balks and start screaming. When you go to them with a study that costs more than $10-15 million, believe me, they're pretty upset with you. And they are also worried that you will never enroll enough subjects. One of the things, epilepsy has gotten bad rap because we've had several trials which didn't enroll sufficient subjects. Finally, lacosamide at that time we didn't have a pediatric dose. They were pushing, but we didn't have a pediatric dose. And we wanted to make sure, one of the big things we've always said is that once our trial is done, it's not just done for adults, but enough is known about children that you don't have to do a separate children trial, which was what happened with ESETT.
Thank you.
Camilo?
Hey, Jaideep. Just was wondering, and thank you for doing the EEG confirmation part, but how are you guys considering cerebral versus complete full EEG in terms of missing seizures from different areas? Also, as you know, the diagnosis of a status electrographic can be very complex and sometimes difficult in a full montage EEG. So how are you guys addressing the definition and deciding if they are in status or not?
So, we are, as I pointed out, we are depending on emergency room staff to apply the leads, right? So getting a full-length EEG, even in medical centers that have 24/7 coverage, within 60 minutes of a patient being enrolled, it seems very, very difficult. And we've tried to test this at places like Michigan or even Stanford, and we don't get a full EEG placement. So cerebral, and we picked cerebral, Joseph, of course, presses me all the time, but Parvizi, but the real thing is that it's the only FDA approved, and they've gone down to age 2 now. So again, it's the only FDA approved device that you can use up to age 2. And how good or bad this data is going to be, Camilo, I don't know, and we are going to find out. And all I can say is I have a very open mind. And as I explained to you, I spent a lot of time thinking, because FDA actually said, why are you doing all the rest of the stuff? Just use your EEG to define whether your treatment worked or not. And I was like, no, I'm not going there because I'll get killed in real world. I know, I have no idea how good the EEG is going to be. So we'll find out. Is all I can say. And we have Eric Rosenthal will lead, Sahar Zafar, Eric Rosenthal, Erika Axine, and who's the fourth person? So everybody's well certified in ICU EEG. Everybody's very well experienced. Let's see what they find out.
Okay, and as we've been talking in the past weeks, I hope you're going to include race as a demographic variability and all that.
So you know that what I want to do this time, I'll try to push it on the side, is at least get zip codes on the patients. So Camilo and I were talking yesterday. We are on this AES task force on healthcare disparities. I don't know if you've seen the paper that there's significant overrepresentation among Black Americans in status epilepticus, and that's not only true from the if you look at Medicare, if you look at medical records data, which we published recently with Gabriela Tentilo at Baylor, if you look at Richmond study, the number of individuals who had status was twice that you would expect from Richmond's population. You look at ESETT population or Rampart population. Again, Rampart and ESETT can be criticized because we enroll a lot of subjects from inner city hospitals, and therefore there may be overrepresentation. But to me, it's very clear that there is disparity. What I struggle to understand is where does it come from? I don't know the underlying cause of disparity, but documenting the disparity is the easy part. Understanding why do we see such huge disparity, I don't know. Chetan, I think you're next.
Sure, yeah, just quick question. Thank you for the excellent talk. Was there a discussion about using brivaracetam over levetiracetam?
Oh, long ago, they tried to sell us Briv, the Kepra people. They even offered to pay us money to do it, part of it. But they didn't have data for kids, and we were very clear that no kids, no go. Briv was the same thing as lacosamide. They wanted to, but that's why we didn't do Briv.
Okay, thank you.
All right. Well, thank you so much again. I think everybody enjoyed that talk. I really appreciate it. And thanks to everybody for joining, and see you next month when Dr. Hopp will be talking to us at that point. All right.
How did I get after Jaideep? This is like Obama saying that he was after Michelle, right?
All
Thank you, Jaideep. That was fabulous.
Thanks.